Chain reaction is a technique used for amplifying sample of DNA fragment in vitro. Discovered by Gary B. Mullis, a Nobel Prize in Chemistry for his work 1993. This process, a particular DNA segment from a mixture of DNA chain is rapidly replicated and producing a large, readily analyzable sample of pieces of DNA. Process is called DNA amplification. PCR itself does not accomplish DNA typing but increases the amount of DNA available for typing. It is used to produce multiple copies of segment from a very limited amount of DNA. Once a sufficient sample has been produced, the pattern of the allele from a limited number of genes is compared with the pattern from the reference sample. A known match conclusively excludes a suspect, but the technique provides less certainty when a match occurs. Procedure. The theory behind PCR is based on a certain aspect of DNA replication. The enzyme DNA polymerase helps to expand a short sequence into a large one or a polymer, but a DNA polymerase needs a single stranded DNA that acts as a template for the synthesis of a new strand. It also requires a small portion of double-stranded DNA to initiate a synthesis primase. Then new DNA strands are synthesized and amplified behind the primer. Requirements for PCR: heat-resistant DNA polymerase, CAC polymerase, primase, short sequence nucleotide designated to bind at the end of the desired DNA fragment. Deoxyribonucleoside triphosphate, equal amount of ATP, DTTP, DCTP, DGTP DNA fragments. 3 steps are involved in the process: denaturation, annealing, and extension. DAE. Denaturation, heating the double-stranded DNA to almost boiling while dissociated and it will become single-stranded. Annealing. Cooling the reaction will cause the primase to pair with a single-stranded template. Annealing. On the small length of double-stranded DNA, the joined primer and template, the polymerase attach and start copying the template. Extension. DNA building blocks complementary to the template are coupled to the primer, making a double-stranded DNA molecule. Each separated DNA can be served as a template for synthesis as long as primer is provided for each strand, and a reaction is cooled to cause the primer to bind. Primers are chosen to flank the region of DNA that is to be amplified. New primer binding sites are generated on each synthesized DNA strand. The cycle of DNA denaturing, primary annealing, and strand synthesis is repeated multiple times, thereby amplification of the target DNA. After heating and cooling, this exponential process yields 2:20 and more than 1,000,000 copies of the target sequence. The process is completely automated with thermocyclers that contain a heating block and microprocessors. The time and temperature can be programmed for repetitive cycles of heating and cooling, elevating manual intervention. Other PCR methods: dot blot, involving a series of DNA probes dictated target sequence such as HLA-DQA, locus in chromosome 6, producing a pattern of colored dot. Amplified fragment length polymorphism, AMPFLP, short random repeats in which the core repeat unit are 3 to 7 base per length. System utilizing mitochondrial DNA, digital DNA typing. STR analyzes the DNA segments of the number of repeats of at 13 core loci. STR analysis is less susceptible to DNA degradation than other amplified fragment length polymorphism. The chance of misidentification in this procedure is 1 in several billion. Application: PCR technique has a virtually limitless application. It enables research to amplify and analyze tiny DNA samples from a variety of sources, ranging from crime scenes to archaeology remains. Limitation: it is too sensitive and tiny amount of contaminant DNA in a sample may become amplified if it includes a DNA sequence complementary to the primase, leading to an erroneous conclusion. PCR can be carried out efficiently if the DNA polymerase owner remains stable through many heating cycles, since denaturation and primer extension often occur at approximately 92 degrees Celsius and 72 degrees Celsius respectively. So a heat-resistant enzyme known as a TAC polymerase derived from a bacteria is used. Criteria to determine the source: DNA testing laboratories use a two-step process to determine if samples are arose from one source. First, DNA binding patterns are compared visually. If binding patterns are sampled in a question do not match a non-DNA sample, exclusion is declared and no further analysis is required. Second, a visualized match is verified by a technique called computer-assisted allele sizing, which is done by computer software. Basically, the calculated size of an apparent match should fall with 2.5% of each other. When samples fall outside the 2.5% window, they should be considered non-matching. If the DNA binding pattern sample cannot be possibly determined but due to technical problem, the result should be considered inconclusive. So, double-stranded DNA, single-stranded DNA, that is denaturation, and as an enzyme is there, polymerase, primase attached to DNA extension, DNA polymerase extends to primase. Repeat. Thank you.